voiceovers throughout the presentation. Please and thank you. In order for HIV to infect a cell, a few things must happen first. The CCR5 domain and the CD4 domain, found on GP120, must bind and transform to the CCR5 receptor and the CD4 receptor found on the cell membrane. Once this occurs, GP41 pierces the cell and the viral RNA is inserted into the cell.
Delta 32 is a deletion mutation that deletes 32 base pairs from a human's gene sequence. A human's gene sequence is composed of the following amino acids: guanine, adenine, thymine, and cytosine. These amino acids form together in groups of 3 to create a codon. The 32 base pairs deleted causes a frame shift mutation because 32 is not divisible by 3. For example, if you have a sentence composed of 3-letter words and delete the A and CAAVS, the newly modified sentence is distinctively different from the original. When this occurs, the CCR5 receptor becomes absent from the surface of the cell.
This is a picture that shows the CCR5 gene sequence. Highlighted in red, you see the delta 32 portion that becomes absent if a person is homozygous for the deletion mutation.
Timothy Ray Brown, also referred to as the Berlin patient, was the first person to have ever been cured of HIV. When he visited Dr. Gerhooter, he had HIV and leukemia. Dr. Hooter found a bone marrow donor that was homozygous for delta 32. He genetically altered these genes and placed them into a vector. The vector was then placed into a cell line. A bone marrow transplant was performed on Timothy Ray Brown, and his HIV status was reversed.
The goal of our project is to develop an easier gene therapy technique in hopes of curing HIV. In order to collect a blood sample, we had our donor fill out an informed consent form. A blood sample was then collected from the donor, who we refer to as AF2. We purified the blood sample to collect the lymphocytes, also known as the white blood cells. The white blood cells were purified to collect the DNA.
Again, you see the CCR5 sequence. Delta 32 is still highlighted in red, and in yellow you see 2 oligonucleotides, Hindi3 and NOT1. These oligos are vital for a proper digestion.
We chose PLN-CX2 as our vector because it is designed for retroviral gene delivery and expression.
The digestion was performed on PLN-CX2 in order to prepare it for a ligation. The digest was performed using Hindi3 and NOT1. Once cut, Hindi3 and NOT1 form sticky ends that will help prepare it for a ligation. A similar digestion was performed on AF2.
In this ligation, digested PLN-CX2 will form with digested AF2. They form recombinant DNA, which will be used in the bacterial transformation.
A bacterial transformation was performed on the ligation of PLN-CX2 AF2. PLN-CX2 is unable to infect U937 cells. Therefore, a transduction was performed to integrate PLN-CX2 AF2 into the cell line PT67, which is capable of infecting the U937 target cells.
We chose U937 cells as our target cells because they possess the CCR5 gene, but they do not express the CCR5 receptor. In the future, we plan to test for the presence of CCR5 in our U937 cells.
Thank you for watching. If you have any questions, feel free to ask using the discussion widget above or the email below.